So the title is about the emergence of a lattice from differentiated cells. So this is definitely a biology talk, specifically a developmental biology talk. But as I say, I direct this NSF Simons National Institute for Theory and Mathematics and Biology, and it's a joint partnership in sponsorship between the NSF and Simons Foundation, and it also is a partnership between Northwestern University and the University of Chicago. Two universities in the Chicago area, we partnered up and to be able to have an inter-university institute that is not only for ourselves, but also for members of the entire community in the United States and around the world. And the whole goal of this institute is to catalyze the better integration of two disciplines that are quite separate from one another, mathematics and biology. And our vision is twofold, actually. One is to drive the understanding of mathematical basis of constraints that drive biological capabilities. So where mathematics helps us to understand biology to a greater extent. And in return, we also want to develop new mathematics that are actually inspired by biology. Now, the basis of constraints, living systems are constrained at many different levels. At the physical level, chemical, historical, through evolution, and biological. There's myriad constraints that limit what living systems can do, but within those constraints, evolution has refined amazing capabilities that living systems can attain. And of course, mathematics is well suited to understanding the nature of constraints and at all levels. And so we think this is a perfect marriage of the two disciplines to understand something about what the NSF calls the rules of life. So one of the big 10 ideas that the NSF has spearheaded for this decade is to understand what are the rules of life. Well, rules in a game are basically constraints that limit the capabilities of the players of a game. Likewise, you could argue that constraints, biological constraints, are defining the rules of life. Now, the way we've organized our institute is not by fields, but basically framed it in terms of concepts. Universal concepts that are shared between mathematics and biology: fidelity and variation, fitness and optimization, information processing, learning and adaptation, prediction and anticipation. And what these different themes provide is an entree for mathematicians to find areas of biology to work in or to develop new mathematics that are inspire them. And these themes or concepts cross all the fields of biology, as you can sort of see here on the left. They also cross all the fields of mathematics, although these are, of course, the different sub-disciplines of mathematics, but it gives you a sense of what the kinds of a certain example of questions that we can ask that feed into all of these, so that we can integrate not only neuroscience with ecologists, but also, for example, dynamical systems with control theory. Now, the NITMB, so that's its acronym, is a hub for both research and convening. It houses both activities. The research is funding internal projects that are internal to University of Chicago and Northwestern, external to other institutions in the United States. Members of the mathematics and biology community can visit and do research at the Institute, and of course training as well. And we're focusing on ambitious and high reward types of research, things that perhaps the NSF doesn't always fund. And at the same time, we have an array of programs that are for convening, to talk about discovery, to talk about education, outreach, and training. Now, in terms of the human resources of the Institute, the long-term participants, that is, these are faculty members and the fellows and postdocs and students of the two universities. But we have an order of magnitude more people from external of the two universities that are visiting as affiliate members, sponsored researchers, visiting scholars, meeting participants, and students. The Institute itself is physically located in the iconic John Hancock Tower, which is a 100-story skyscraper in downtown Chicago. It's strategically located halfway between the main campuses of these two universities. And of course, it's easily accessible for those who are visiting, who can fly in or travel and stay in the local Chicago area. We occupy most of the 35th floor, and these are photographs. These are not AI taken photographs. These are actual photographs of the very first day of occupancy when we had, that was 2 weeks ago, in which we've got, not all the furniture has arrived yet, but you can see that we have expansive views of both Lake Michigan and the cityscape at this institute. And so if you want to learn more about the Institute, you can visit that website, NITMB.org, and please consider visiting us. It's very simple, low barriers to applying for visiting scholars, or just come whenever you visit Chicago. We'd love to have you visit us. Okay, so the main science of this talk is going to be about pattern formation. And pattern formation in multicellular organisms. So what do I mean by pattern formation? Well, you can think of, for example, the overall body plan as a pattern, right? You have your head to a tail, back to a belly, appendages, and so forth, right? And so there's sort of a gross level pattern that is inherent in every multicellular organism. But also there are patterns that are at a finer scale. So for example, the fur or feathers of these two animals, you can see that they form these patterns that are highly regular in nature for the camouflage purposes. And so at many different scales, both at the whole organism scale, tissue scale, and organ scale, patterns are always emerging and are present in organisms to allow them to survive and thrive. And the remarkable thing about these patterns is that they develop from a single fertilized egg cell. It is very, very simple in structure with radial symmetry. And through this magical process of development, the pattern emerges through both growth and development. Now, this process of development has been studied for a long time, thousands of years. Aristotle is the first to record his study of development. And that was very descriptive, where they basically were looking at the process of these cells undergoing their transformation to form this in real time and recording basically the pathway by which that occurs. But underlying those morphological changes are obviously mechanisms at the chemical and physical levels that are controlling and regulating that process. And this, of course, has been a subject of interest for a long time. And it's an example where, actually, in biology, theory preceded experiment in terms of discovery of ideas about how that occurs. And the person that spearheaded that was a mathematician, Alan Turing. So shortly before he passed away, he published a short article where he was asking the question, how might a pattern form in a developing system? And he came up with a concept that was chemical in nature. He said, suppose there's a chemical that can diffuse to form a concentration gradient from one side to the other side, say, of the inherent structure that's going to be patterned, and that the cells locally sense the concentration of the morphogen and then make decisions about what they should do that are based on that local concentration. And he termed such chemicals morphogens. Now, this idea sat for over 10 years. And the idea was picked up by two mathematicians, Hans Meinhardt and Alfred Geyer, and a biologist, Louis Wolpert, who in the late 60s independently devised or elaborated upon this concept of a morphogen. The idea they came up with was the following, which was, suppose that the cells are indeed reading their local concentration, but they respond in a unified or unitary manner, an invariant manner, whether with a threshold at which above this concentration, they all do the same thing. So let's say they turn into black cells. And then cells that are locally sense the concentration in this range will adopt a different fate, yellow cells. And in that range, they will adopt yet a different fate, red cells. And what does this create? It creates the flag of Belgium. So you generate a simple pattern, and yet it comes from a single source, that is the chemical. And it wasn't until the late 1980s, and people were looking, this was a beautiful, simple idea, and so developmental biologists searched for evidence for such morphogens that worked in such ways, but it wasn't until the late 1980s that the first morphogen was found in the fruit fly Drosophila melanogaster, and the morphogen is a protein called bicoid. And here is a micrograph of bicoid protein, where the level of the intensity of the signal, which is in these spots, and these spots represent nuclei within the developing embryo. You can see that it forms a concentration gradient highest at this end and lowest at this end, because it's basically synthesized over here and then diffuses. And indeed, bicoid is a transcription factor that regulates the gene expression of other genes, including these genes, hunchback and caudal. Now, these are very silly names. Most people outside of Drosophila research find Drosophila genetic names rather strange. But the names are basically named after the phenotypes when these genes are mutated. But nevertheless, hunchback is expressed in this domain here above a threshold, whereas caudal is expressed in this domain here. And shown over here is the expression of hunchback, caudal, and another gene, and you can see it forms three domains. Not the Belgian flag, but these cells are fated to become the head, the thorax, and the abdomen, okay? Which is the basic body plan of the insect. So this was the first evidence of the existence of a morphogen that did something that Meinhardt, Geyer, and Turing also believed. Now, I mentioned earlier in the talk that there is also pattern formation that is on a finer scale. And very often you can see regularity, a repetitive pattern, and it's ordered in a regular array. So for example, the whiskers on the face of a cat, the hair cells in the inner ear of a mouse, the retinal cells, the photoreceptor cells in a zebrafish eye, and these chemosensory bristles on the legs of a fruit fly. All of these are highly ordered and repetitive. And that's another interesting problem. Like from a disordered cellular milieu, how do you generate a regular pattern? And again, we can come back to Alan Turing, because Turing elaborated on his idea of a morphogen to come up with the reaction-diffusion model, where it wasn't just one molecule or chemical, but two. There's one molecule that reacts with another and vice versa. This molecule inhibits this molecule, whereas this molecule activates itself and it activates the inhibitor. And these are local reactions, and yet they are also free to diffuse globally through space. And so at different positions within the structure we're talking about, there's different concentrations of activator and inhibitor, and that generates, based on these reactions, different kinds of responses. And Turing developed PDEs to describe this, where the activator is U and the inhibitor is V. Here's just a straightforward example of that. They're basically the terms of synthesis, decay, and diffusion. And one can structure these functions in a variety of different ways that fulfill this basic architecture. And one can derive different outcomes based on that. The outcomes are also dependent on the choices of the parameters that are involved with this. And what Turing had found when he was playing around with this in the 1950s was that when he found that when the inhibitor had a diffusion coefficient that was much, much greater than the diffusion coefficient for the activator, it produced some very, very interesting results. These are simulations not done by Turing, obviously, but much later, but show you examples of those two conditions where the inhibitor is much more diffusible than the activator. And what it produces is either these moving waves that he called type 5 patterns, or these standing waves, which he called type 6 patterns, and now other people call them Turing patterns. And this is an interesting one, which is actually a hybrid between the type 5 and type 6. So with this very simple system, if you can find the right parameter values and the right construction of the function itself, you can generate all sorts of complexity in terms of patterns. And this, of course, was also of great fascination to biologists who were interested in the creation of patterns that have a lot of regularity. Because depending on how you tune the parameters, you can get a variety of different standing waves, the type 6 patterns that emerge. And in fact, you can use that to actually simulate the natural patterns that are found on objects such as these seashells or on the skin of these tropical fish. On the right is basically a reaction-diffusion simulation that matches the natural pattern. So despite this really beautiful and powerful system, it's been very challenging to basically provide experimental evidence that Turing mechanisms such as this actually generate the vast variety of patterns that we see. I think there's a question at the back.
Yeah, so you had a nonlinear term in your equation. Is that fundamental to all the patterns?
I believe so, yep. Now, to create the patterns that are either type 5 or type 6, the diffusion coefficients have to be very different in value. So other types of patterns will emerge if the diffusion coefficients are altered to, say, be more equal. So you can have traveling waves, but you can also have spots that come up and come down. There's all sorts of patterns that one can create. So, as I say, providing experimental evidence to support this idea is much more challenging than it has been to support the idea of morphogens. And one of the reasons is that to be able to go in and manipulate, experimentally manipulate the system in a manner which is predicted by the reaction-diffusion model and compare the experimental outcome to prediction is very, very challenging in biology, at least in the current day. So let me give you an example of how a Turing-type mechanism being very seductive can lead biologists down a path in which they find that actually it's not as simple as that. And that is a study that we and many others have taken to understand the pattern formation of the compound eye of fruit flies, which is, of course, a very regular pattern. Ramon Cajal, the famous neuroscientist, thought it was the most beautiful and complex pattern in all of the various systems he studied. if you cut away through those lenses, you see this beautiful pattern of these groups or clusters of dark objects that form these trapezoids. And note that there's chirality to the trapezoids and mirror symmetry. And this dividing line, which goes along the midline of the eye. Each of these clusters, each of these spots represents a photoreceptor cell. And so these are the organelles that are responsible for basically capturing the photons and then sending that information to the brain to allow the animal to perceive its visual environment. But you can see the beautiful regularity of this structure. And if you look for errors in it, they're less than 0.1%. And yet that beautiful, precise pattern develops from a rather disordered epithelial tissue that is created during the embryo, when the embryo is developing, and then is set inside the body of the animal, deep inside the body of the animal, as these imaginal discs, which are simple epithelia. And the eye epithelium is this structure here, and this is a scanning electron micrograph showing you one such eye disc attached to an antennal disc, and it's got an optic stalk that connects to the brain, and this is where the axons of the photoreceptors move to innervate the underlying brain. Now, if you take this structure, you'll find that it's actually composed of two epithelia, the disc proper, which contains the eye or the retina, and then a squamous or thin layer epithelium that if you tear it away, as if it was a birthday present, you find underneath this vertical furrow. And when people discovered this furrow, they coined it the morphogenetic furrow. And if you take the eye at, say, an immature larva, the furrow would be located over here. And as the larva matures, the furrow is at a different position in this eye field, leading people to realize that this furrow moves across the eye. And experiments determined that it was basically moving at a constant speed, or they were inferring that it moved at a constant speed. Now, this furrow actually coincides with a moving wave of gene regulation. And so shown in here is one particular gene, highlighted in green, that is being expressed in the cells that are within that furrow. And you can see that it's a moving front in which basically is creating the pattern behind it. So the green wave moves from this end to this side at a constant velocity, and on the trailing side, on the wake of the wave, the pattern of these photoreceptor clusters emerges. Now, how does this work from a genetics perspective? Decades of research has uncovered basically many molecules that are involved in this process, and I'm only showing you a small subset of these molecules with these different names. Some of these molecules are involved in signaling from one cell to its neighbors, much like a morphogen might do. So hedgehog, for example, diffuses and activates cells here to make this morphogen, which is DPP, which then diffuses and puts these cells into cell cycle arrest and prepares them for the transmission of the furrow as it passes across the eye. And a key target of these signaling pathways is this gene called atonal, which encodes a transcription factor, basically telling the cell that it's proneural. That is, it has the potential to become a neuron, a photoreceptor. But only a subset of those cells that are informed by atonal to be a photoreceptor actually do become a photoreceptor. And the vast majority of them basically relax to an undifferentiated state, similar to the state they were in before the furrow passes. But going through this transitional process of cell states, there is basically a pattern of columns of these R8 cells that emerge on the sort of right side of the furrow as it's passing across from right to left. And the way that people thought that this periodic pattern of R8s emerged was through something like a crystal. That there's basically the position of the emerged columns, emerged R8 cells will instruct where the next R8 cells will actually come. And in fact, Seymour Benzer, who pioneered the development or the study of Drosophila eye development, called this a neurocrystalline lattice. Now, if we just look at the basic architecture of how this works, atonal, as I say, makes a cell proneural, and it activates two long-range inhibitors, or they're thought to be long-range inhibitors. One protein called scabrous and one protein called EGFR. These long-range inhibitors inhibit atonal, which activates itself. So you can see this has the basic architecture of a reaction-diffusion model. And the idea is that simply you convert this stripe of cells into periodic clusters of cells that are atonal positive, and then a different mechanism involving short-range signaling through a different set of ligands and receptors will resolve these groups or clusters of proneural cells to a single cell, which is going to be the R8 cell. But the key is that there's the potential for a reaction-diffusion mechanism to be at play here through the action of atonal and these long-range inhibitors. And so Vasile Alba came up with an idea to model this in silico using Turing modeling. And this was inspired by a model that was developed by Francis Corson from the École Normale Supérieure. And basically, it involves not sort of diffusion coefficients, but signaling that is modified depending on distance between cells and has this simple structure here. And if you follow what it can do, this is the leading front of atonal expression, much like the morphogenetic furrow. And you get a very nice regular pattern of intermediate groups or clusters of atonal positive cells. And this particular model is very robust. It's robust to the speed of the wave. It's robust to the disorder of the tissue. It's robust to the parameter value, many of the parameter values involved in the modeling itself. So there we have a plausible explanation for how this is created using a simple biochemical circuit such as this. But there's one nagging issue here, which is that it's a furrow. So there's a mechanical buckling of the tissue itself. There's a mechanical wave. And how is this generated? So this is showing the profiles of individual cells, and you can see that they sort of order in irregular polygons. Some of them are more circular, like this. These are actually cells that are undergoing cell division. But these irregular polygons have different areas. So over here and over here, their areas are roughly equivalent. And you can see the areas of the polygons here are much smaller. So what's happening is that this top layer of the tissue, the cells are constricting in area. And that then naturally causes a buckling of the tissue, and that is responsible for the furrow. So you have basically this existing tile work of cells that we'll call in a relaxed state. They constrict, and then the vast majority of them relax again to that dilated state, with the exception of these clusters of cells that are actually the photoreceptors. So the photoreceptors never relax, but remain apically constricted. So the biochemical model is sufficient to explain the generation of this regular lattice, and yet it doesn't explain why there is this mechanical wave that is coincident with the chemical wave. So a graduate student in my lab, roughly 8 years ago, Kevin Gallagher, decided to do live imaging of eye disc development. And you might think, well, that's rather trivial because you can just make a movie. But people have been trying to do this since the 1960s, virtually everybody working in eye development, without success. Of course, you can't image within the body of the animal because remember, the eye disc is buried deep inside of the larva. And so you have to surgically excise the tissue. And as soon as you do that, people found over and over again that basically the eye would stop developing, in some cases die. So Kevin spent basically 1.5 years working out a method to be able to get robust eye development to occur ex vivo. And it involves surgical excision of the eye disc and placing it in basically a dish with media and mechanical support, and then visualizing by light microscopy, fluorescence light microscopy, to visualize particular fluorescent proteins within the living tissue. And then a lot of image processing with machine learning, pixel classification, surface detection of the signal itself, and then segmentation and tracking. And this part also required a great deal of innovation on Kevin's part, which he did all himself. And so here is one of Kevin's movies. I don't know if the lights can be turned down briefly for this. So what we're looking at here, the signal is green fluorescent protein, which actually comes from a jellyfish, and it's fused to E-cadherin, which is the adhesion protein that is present on the sort of top surface of the cell, the apical region of the cell. Let's run this again. So you can see that the furrow is moving. There looks like sometimes these bubblings, and these are cell divisions that are occurring. But you can see that as the furrow moves, that these buds of small constricted cells are emerging off of the furrow, breaking off, and then going free. So one of the things that we expected to see was the following, which was that the furrow moved like a compression wave, that cells did not move relative to each other. And this assumption was based on experiments from the 1970s using clones of cells, basically lineage-related cells, sister cells, and then finding those sister cells remained together in the final tissue, implying that there was no great large-scale migration of one cell relative to the other. So the field has always assumed, just like with the Turing model that you saw with the vertex model that Vasile built, that the cells are not moving relative to each other. They're staying still. And that would imply that there's a prepattern of cells which ahead of the furrow, before the furrow's even passed, these cells are fated to become proneural and R8 cells, just based on their position relative to everything else there, because nothing is moving around. Everything is stationary. But in fact, we saw at the very beginning of our analysis something that was not like that at all, that there was cell migration or cell flow where cells were moving relative to one another. Not great distances, but large enough distances. And so each of these arrows represents a vector of cell movement over average time. Here's the furrow here, which is marked by these cells that are in red. And you can see that there are these periodic clusters of cells that are together that move with high velocity beside clusters that are moving with low velocity. And they oscillate. Also clusters here that are moving in the opposite direction.
How many cell diameters?
Where they basically are shared, about 10.
10 cell diameters.
Yeah. If you measure the period length of those by Fourier transform analysis, the period length of the stable period length is the same as the distance between one of these clusters and its neighbor over here. Okay? And you can sort of see that here. Here's a high velocity group. Here's a high velocity group. They kind of match the distance there. Okay? So the scaling of these are, the period length is scaled quite closely to the period length of the groups. And as I said, they oscillate over time. So here, a high velocity cluster, 2 hours later will be lower velocity, and then it goes high, and then it goes low. It's kind of dissipating over time here because the furrow actually does slow down somewhat within these explants sometimes. But here, there's a cluster of cells right beside this high velocity, that's low velocity, 2 hours later. Now it's high, low, high. And so you have these oscillations of cells that are high and low that are basically out of phase with clusters that are beside them that are the opposite. So then obviously, there is no prepattern that looks like a hexagonal lattice ahead of the furrow, right? Like I showed you in that schematic, because the cells are moving. And they're moving in a very complicated way. So then what is the prepattern? So this is the prepattern here. So what Kevin has done has identified all the cells that are going to form clusters and colored them different colors. So here's a cluster that is in the one column, and here's another green cluster here. These belong to the same column. So green is one column, purple is a different column, yellow is a different one, orange, etc. So these are in different columns, and yet you can, and that column forms a lattice pattern in the final form, but you can see that the prepattern actually is not like that at all. The prepattern, at least in the furrow, almost resembles a rectangular lattice, not a hexagonal lattice. And then you can see how that hexagonal or that rectangular lattice then evolves as these clusters emerge and stop moving relative to the furrow. The furrow is constantly moving, but they stop moving. And they do so in an alternating manner. So there, the yellow stop moving, the orange and the blue are still moving, and the orange stops moving, and then the blue stops moving. So you have this basically movement, constant velocity of these groups of cells that are fated to become the actual photoreceptors. And at specific intervals of time that are 2 hours apart, the clusters will stop moving. And that's shown here with this distribution, 2D distribution. You can see a bimodal distribution of cells that are moving and not moving in that region. Yeah.
So there is a static field of half. Yeah, I pressed it. So there is a static half space of cells in front of the furrow. The furrow comes. Then there is this nonlinear reaction-diffusion system that breaks the symmetry, right? And specifies the. So once you have a group of cells, this little band, it acquires velocity in the posterior direction. Correct?
In the posterior, which is to the right.
Yeah,
Yes.
yeah, in the posterior direction.
Yes.
And it moves for a little bit while rearranging.
Yes.
Is that the picture?
Yes. So there's movement both in the anterior direction and the posterior direction. So you'll see with this actually quite visually, the movement, and then more analytically.
Thank you.
So what we've done here is basically made vertices. These vertices represent the future R8 cells in a cluster. Okay? And so they're all just linked together to form a lattice. And you can see in the furrow here, the lattice pattern is roughly a rectangular lattice. And now you can watch and you can actually see the velocities of the groups of cells, how this one is stopped, but this group is moving, and so that stretches that apart. And you're starting to make triangles that are more isosceles. But then you can see posterior movement, for example, right there, which is stretching them even further. And so these organized flows of cells that are not just in the furrow, but also posterior to the furrow, basically derive that or evolve that rectangular lattice into a triangular lattice, a regular triangular lattice. Those are individual R8 cells. There are others. This is just for visualization of how the lattice evolves. All right. So you have this very complicated flow of cells. What's generating these mechanics? And the clue came by analyzing this is a one-dimensional profile of the average anterior velocity relative to the furrow, of cells relative to the furrow, anterior to posterior, over all the frames of a movie, over all the cells in a specimen, over all of our samples, replicate samples. And just to highlight how reproducible this pattern is of flow, this is the moving line average and the 95% confidence intervals shown right here, okay? So it's highly reproducible. And the important thing to keep in mind is that you can see that here's the furrow positioned here, that on one side of the furrow, the cells on average are not moving either direction, posterior or anterior, and likewise here. So those are static points. But then cells are moving in directions suggesting that there's actually a source of cell flow coming from here, because cells are flowing this way when they're on this side, and they're flowing that way when they're on this side of this transition point. And the same thing here. Cells are flowing towards this point when they're positioned here, and they're flowing towards this point when they're positioned here relative to this. So this behaves like a sink. So it suggests basically there are zones which are sources and sinks of cell flow. Yes, at the back.
Begin differentiating before they end up in the cluster?
It's a great question. So the R8 cell is the first to differentiate. And it is basically, think of it as a pioneer or a founder for the cluster. So as a founder, it founds all the rest of the photoreceptors in that cluster. So it's the first to differentiate. And then very shortly thereafter, what it does is it locally signals some of its neighbors to tell them that they should also be photoreceptors. And so then you get this formation of the 5-cell grouping. But the actual positioning of those clusters is dependent on where the R8 cell, which is the founder, is positioned. Does that help answer your question? Okay, so there's evidence that there's a source and a sink of cell flow that has a very fixed position relative to the position of the morphogenetic furrow. And what causes this source and sink? Well, these points of sink and source coincide with the same place where the cells are maximally changing in their area or volume. So for example, at this point where the sink is, this is the area where there's maximal contraction of the cells. Whereas over here, at the source, this is the area of maximal dilation of the cells. And so if you think about the cells are shrinking or expanding at the highest rate at these points of source and sink, that would suggest, it suggested to us that there's a pressure gradient that is created by the contraction of the cells that then by their shrinkage causes flow towards them, and expansion of the cells pushes the other cells that adhere to them away. And therefore, there's a pressure gradient which is generated by the positioning of this maximal expansion and contraction of the cells, so that there's a pressure gradient. But that wouldn't explain why there's a periodicity to the cell flows within the morphogenetic furrow. So one model would be that there's a pressure gradient that is produced, but it's got periodicity to it itself. So there's a zone of high pressure, a zone of high pressure, so you get maximal flow away from these zones, less here. But you would then expect to see some kind of periodicity of cell flow on the posterior side, and we didn't see any evidence for that. So in the far posterior, the cell flows are not bimodal, but follow a normal distribution. Instead, what we think might be going on is that there is something that occurs very much like fluids that have to basically pass over an immobile object. And when they do so, of course, there's high flow rates surrounding them and low flow rates in the wake of the immobile object. In the context of this developing eye, what might these immobile objects be? Well, these clusters themselves could act as immobile objects. If they stopped moving altogether and resisted the flow, then one could imagine that they would then focus and deflect the cells surrounding them to go at a higher, slower rate. And when we looked at basically the velocity at which these R8 cells go, they're very different from the average or non-retinal cells. They basically stop flowing shortly after they emerge from the furrow. And they stay basically immobilized at zero velocity until all the other cells catch up to them at this point of the source of cell flow. And then they let go, and it looks like they lose their anchor, and they start to flow with the ambient cells that are surrounding them. But for that brief period of time, when they act as immobile objects, we think that might be sufficient to deflect the flow and create this periodicity. Okay, so these are our observations, and it's just a summary of them. We have the furrow itself here straddled by a sink and a source of cell flow we think is created by a pressure gradient. That gradient then causes periodicity due to the immobilization of these clusters at these particular locations, and then posterior to that, everything flows away. Now, it's challenging from a biological perspective to be able to actually use methods to sense whether there actually is a pressure gradient. So unlike a lot of inanimate systems, like in physics or engineering, where one could actually have instrumentation to measure such things, in biology, alas, we have wonderful tools, but that is very challenging. So if you have questions about whether we can actually show that there is a pressure gradient using sort of those more traditional methods, the answer is not yet. But one would predict that if you impaired the flow somehow by reducing, say, the dilation of the cells, inhibiting the dilation of the cells, that one would not generate this lattice pattern, hexagonal pattern, but would maintain the rectangular lattice pattern. And the one thing in biology that we have is genetics. And genetics is very powerful because you can generate a mutation in a very critical gene that does one specific thing, let's say causes cells to dilate. And the consequence of that is perfectly surgical in that there is no going in mechanically and perturbing it, but literally the animal is engineering the perturbation itself naturally. And we found one such gene that showed such effects, and that was scabrous. So recall that I talked about the Turing reaction-diffusion model invoking scabrous as a long-range inhibitor. That was based on its genetic phenotypes without ever being able to look at these sort of properties of mechanical flow in a scabrous mutant, but now we were able to. And what we discovered was that the flow in a scabrous mutant was non-periodic in the furrow, right? There's the Fourier transforms, and was very, very weak. So here is the, in red is the scabrous mutant distribution of anterior velocity, and in wild type is the black-gray. You can see the bimodal distribution, fast flow, low flow, and you see there's no periodicity, and in fact, all the cells are behaving with low flow. So they're not flowing very fast. Why is that? Well, they still have a sink and a source of cell flow that straddles the furrow. It's a little bit closer. The source, you can see that the flow rate over here is equivalent to wild type. The flow in the furrow itself is reduced. The flow away from the source on the posterior side is also reduced, as if the source was not as strong at pushing the cells away, and so they were not moving as fast away from the source. And indeed, when we measured the rate of change in basically the dilation of these scabrous mutant cells, specifically at the transition point, right, where the source is located, that it's greatly reduced in a scabrous mutant. In other words, the scabrous mutant cells are not dilating as quickly as normal, but about half as quickly. And that seems to be causing an aberration in the flow rate of the cells and also an aberration in generating periodicity in the flow. So what effect does this have on pattern formation? So this is scabrous, where each of these groups of cells represents a photoreceptor cluster. And what I think you can see, this is the beginning of the movie. So here's the prepattern. You can see it's very, very disordered, but roughly a rectangular lattice. And the same thing here, a rectangular lattice. So you don't see the hexagonal lattice pattern, but you see something that looks more like a rectangular lattice and the fusions of these groups of cells, right? And if you just watch how it evolves over time, you can see they emerge at the right time, and the spacing between this column and this one column over here is about right, but the spacing between individuals are incorrect, and you instead get what looks like the rectangular lattice pattern. So in other words, the rectangular lattice pattern is a prepattern that is established independent of the cell flow mechanism. But the cell flow mechanism is necessary for that rectangular lattice pattern to evolve into a hexagonal lattice pattern. And so this is our current model, where we think that the prepattern here is still established by interactions between atonal and Spitz EGFR, could still represent a reaction diffusion mechanism that's biochemical in nature, but doesn't generate the hexagonal lattice on its own, but is sufficient to generate this rectangular lattice. That then through this furrowing, which generates, because you have a furrow, you have to shrink cells ahead of the furrow and expand them in front of the furrow. That creates the buckling because you have to have them small in the furrow. And just by nature of changing that is sufficient to generate a pressure gradient that can generate cell flows. And then that can evolve the rectangular lattice pattern into something that is hexagonal. So this is an example from one tissue, the compound eye of a fruit fly. Is there evidence for this kind of coupling of a chemical and a mechanical system in other sorts of organisms, in other animals? And the answer is yes. So for example, the developing central nervous system of the early frog embryo involves a similar kind of wave of neural induction, that is biochemical induction of neural identity that is coupled with a furrowing in the early embryo. The formation of the regular follicular pattern on birds, right, where the feathers are going to be placed, ultimately is a hexagonal pattern, but also works by a coupling of a wave of local cell-cell signaling with mechanics. The inner ear of a mouse, similar thing, a mechanical wave that is coupled to a moving front of gene regulation. And in the central nervous system of the optic ganglion that actually feeds, the eye feeds into, a similar kind of phenomenon. So it is possible that this kind of mechanism where one combines both biochemical mechanisms such as a reaction diffusion model or others involving morphogens, can work hand in hand with mechanical mechanisms to generate much more elaborate and perhaps more robust patterns that we see throughout the living world. And so with that, I'd like to conclude. Kevin Gallagher did all of the experimental work. Vasile Alba, who was a postdoc with Madhav Mani at Northwestern University, did the mathematical modeling. And this work was funded by the National Institute for Theory and our precursor center, which was the NSF Simons Center for Quantitative Biology. And with that, I'd be happy to take questions.
Fascinating talk. Any questions? Many questions, please.
It discussed is occurring mostly in surfaces. I was wondering if it's generalizable to 3 dimensions.
Yes, it has to be generalized. This has to be generalizable to 3 dimensions because the epithelium is 3-dimensional. What you're only looking at is a 2-dimensional layer of it, which is the top layer, the apical layer. What's underlying that, of course, is the middle layer where the nucleus is located, where the transcription factor, atonal, is working, plus others. And of course, the basal layer, which is adhered to the extracellular matrix. And one of the limitations of this kind of visualization is we cannot be able to see the entire cell in 3 dimensions. If we could, it would be wonderful, but in the current technology we have, we can. Now, we are working on being able to visualize dynamics of protein expression, say, for example, atonal in the nucleus. And then another group in France is studying how these cells actually move relative to one another on the bottom layer of the tissue. And so perhaps altogether, we're going to get a much better construction of it. So I'm only showing you the top layer and showing this. What's going on down here? Are they moving along? Are they not? We don't know yet.
All right, yes, please.
As far as your institute, it sounds
really exciting, except I would like to see artists involved in that, because they can come out of the box and they have something that you're not thinking about. Being a recovering artist myself,
All right.
What,
Thank you.
in what ways would you suggest artists could have an impact?
just mill around.
All right. One more question. Yes, please.
If I may, so in your patterns, I filmed Benoit Mandelbrot in a snowstorm, and he could visualize patterns, but your patterns are hexagonal. I'm just wondering, have you thought about the fractals?
Fractal patterns. We don't see any evidence for fractal patterns in this system, but certainly in lots of other biological systems, there's evidence of fractal patterns. Vascular systems, in particular, in plants and animals, they have fractal properties, and you can see that with 1/f behavior in quite a few number of systems. So there is fractility in living systems. We haven't seen it here, but not yet.
All right, so we're a little bit over time. One more question. Yes, please.
So, hi. I was wondering if you could comment on what would be the sort of next chemical level. So there's presumably adhesion that's preventing the cells from moving and that's making them adhere to each other as they come out. How do you think that fits into, is that a precursor to the change in the flow rates? Or is that, something's stopping and then the flow is going this way? Could you comment on that?
So all the cells remain adherent to one another, although in order for, say, high cluster to be beside a low cluster, at that boundary, cells have to change neighbors, and they change neighbors locally. To be able to change neighbors, they basically have to momentarily let go and latch on to a new one. They never actually, bud off completely. And you could think of it almost like, because there's an adhesion that is slowing it down, it's like a drag. And so, for example, there's ideas about how would the drag be influencing the flow rates, for example. We see, for example, those clusters form as lines, vertical lines, you probably noticed roughly 5 cells like this, and then they arc, and then they buckle into a rosette. And we're really interested in that, and we think the buckling process might be the drag force of the cells flowing around the immobile pinned object and causing all the other cells just to physically fold in through a drag force mechanism. We have no idea. It could be pinned from below, right? Coming back to your question, it could be pinned from above. We have no idea.